Hello and welcome to the Introduction to Biology unit. This unit contains a lot of various information regarding what biology is, how we classify living things, how we identify something as being a living thing, and also a brief study of scientific experimentation. So let's get started. Our first note is life is everywhere. Biology is defined as the scientific study of life. Bio means living, and ology refers to the study of, so this is where the definition comes from. Biology is influenced by many factors. Some of those are species interactions, and what we mean by that is things like predator-prey, relationships where both species may benefit from each other, such as mutualism, and so forth. The next one is climate, disease, pollution, and also population size. So we'll be investigating a few of these factors throughout the course, and so you'll hear from some of these again. Our next big note is characteristics of life. So we're going to list several characteristics of life, and what this means is these are things that living things must possess in order to be considered living. So the first one is they have to be organized. Life is organized, and the basic unit of life is the cell. Now, the cell is the smallest functional unit of any living thing and forms the basis of higher levels. Every organism consists of one or more cells. If an organism is made up of one cell, we call it unicellular. If an organism is made up of more than one cell, we define it as multicellular. Now, while the cell is the basic unit of life, we know that cells are made up of smaller components called organelles, and organelles are made up of even smaller components called molecules, and even smaller than that are atoms. So while the cell is the basic unit of life, the atom is the smallest particle of all matter, of all living and non-living things. Atoms will then bond together to form molecules. Molecules, as you can see in this graphic, will bond together to form large molecules. And the word that we use for large molecules is macromolecules. So we can see here we've started with atom, and we said atoms are bonded together to form molecules. Molecules are bonded together to form big molecules called macromolecules. And then those make up organelles. Organelles are the parts of a cell that each have different functions and help the cell to be able to maintain itself. So organelles make up cells. Cells make up tissues. And so a tissue is defined as a group of cells that have a common function. If we put several different tissues together that work together, we call that an organ. If we have several organs functioning together, we call that a system. And all of the systems working together form the organism. Now, organisms of the same species can be grouped together and are called populations. So if we talk about all of the humans in your city, then we are talking about a population. These are members of the same species living in the same place at the same time. If we're talking about the number of earthworms that happen to live out in your yard, we're talking about a population. If we're talking about the number of robins that live in your yard, then we're talking about a population. But when we group all of those together, the humans, the earthworms, and the robins, then we're looking at a group called a community. Communities are made up of different species that live in the same place at the same time. So while populations are the same species, groups of populations would indicate a group of different species living together, and that's called a community. If we combine the community with all of the living and non-living factors of their environment, we're looking at an ecosystem. So we're looking at things like temperature, we're looking at things like soil content, amount of precipitation, along with all of the species that happen to live there. Sometimes ecosystems can be grouped together with similar other ecosystems, and we call those biomes. And then biomes are grouped together and they make up the entire biosphere. The biosphere is the area where we find living things, and this can extend down into the earth and even up into the atmosphere for several kilometers. So this is a big infographic here that shows you all of the levels of organization. I would encourage you to especially focus on this middle section here, these that involve the organization of living things. I would be sure to know those, that cells make up tissues, tissues make up organs, organs make up systems, and systems make up organisms. And we can go smaller than that with organelles all the way down to atoms, and we can go larger than that on a macroscopic scale all the way up to biosphere. So number one, all living things have to be organized. Secondly, all living things require energy. So in your body, you have a lot of chemical reactions going on. Some of those reactions are going to break down substances like the foods that we eat, while others are going to build up substances. They would take the atoms from the foods that we eat and rearrange them to form the molecules that we need. The sum of all of these chemical reactions that take place in your cells is called metabolism. So these reactions allow you to acquire nutrients, use the energy obtained from them to build those new structures, repair old ones, and even reproduce. There are several ways that organisms obtain their energy. You probably have heard of a producer. A producer is an organism that can make its own food by extracting energy from other sources. And one example of this that you may be familiar with is photosynthesis. So plants and other microbes can undergo photosynthesis to create their own energy. So another word for a producer is an autotroph. You may not be as familiar with that term. So an autotroph is another word for a producer. Secondly, there are organisms that cannot make their own food, and so they have to consume other organisms. And this is where we come in. We fall in this category called consumers. We eat our food, we ingest our food, and those molecules are then used to supply energy and nutrients to all of our cells. Another word for a consumer is a heterotroph. So autotrophs are producers, and then heterotrophs are consumers. Another example of a heterotroph is an amoeba. You can see an amoeba diagram here on the right. Amoeba is sort of like a blob. It doesn't really have a specific shape. And these extensions of his cytoplasm that you see here, these are called pseudopods. And he uses these to surround his prey, and then he takes them into the cell body using a vacuole. So in this way, he is consuming other organisms, and so this is why he is a consumer. A subcategory of a consumer includes decomposers. You may be familiar with fungi, like mushrooms, that break things down, and they return the nutrients of whatever it is breaking down back into the soil. So I like to call them nature's garbage men for this reason. They kind of recycle those nutrients. You'll find fungi on things that are like rotting logs and so forth. So it's helping to decompose that structure and return the nutrients back into the environment. So because it is sort of consuming another organism, then fungi are considered heterotrophs as well. So all organisms do have to obtain energy in one way or another. Third, life maintains homeostasis. And homeostasis is just a big word that means that we're going to keep all of the levels where they should be. So it literally means steady state. It is the maintenance of a stable or constant internal environment. An example of this would be internal body temperature. Your body temperature should be somewhere around 98.6 degrees Fahrenheit or 37 degrees Celsius. Those are the same. And so your body works to sort of maintain that on its own. So we have what's called internal homeostasis. Your body will, if you get cold, your muscles begin to shiver. Your hair on your arms and your head kind of stand up a little bit to try to provide more insulation. So if you get too hot, then your body sweats and the sweat evaporates to try to cool your body down. And so those are all ways that your body helps to maintain homeostasis. Some organisms, like reptiles, though, don't have that internal homeostasis as complex of a system as we do, so they have to kind of work to maintain their homeostasis. You may have seen turtles sunning out on a rock, and the reason that they do this is to warm their body up. And when they get too warm, then they will jump off into the body of water to cool back down. So some organisms have to maintain it externally. Fourth, life must reproduce. There are two basic types of reproduction, sexual and asexual. And the main difference between these two is that in sexual reproduction, you have genetic material from two parents that are combining, but in asexual reproduction, there's only one parent. So for sexual reproduction, you may have the genetic material from the male sperm uniting with the female egg. And so half of the genetic material comes from the female and half of the genetic material would come from the male. And you get that new combination of genetic traits. This will increase genetic diversity in the species. Because you, for example, are not a carbon copy of either of your parents. You're a combination of the genes that they contain. In asexual reproduction, though, because there is only one parent, there's only one place that the DNA can come from. So the offspring are going to be identical to that parent. Now, examples of this would include binary fission in bacteria. Binary fission is a very quick form of reproduction in bacteria. It's where the bacteria makes a copy of its genome, of its DNA, and then it basically just splits into two cells, each cell receiving a full copy of the genetic makeup. Another example of this is vegetative propagation, and this is seen in strawberries. Vegetative propagation is a big word. We call this runners. If you look right here, where I'm circling here in yellow, this is a runner from the strawberry plant. And when that runner touches the soil, it will root and form a new strawberry plant. Because there's only one parent plant in this case, this is asexual reproduction. Another example is in fungi. Fungi produce spores, and spores are tiny little reproductive cells that when they land in a favorable environment will begin to grow a new organism. So in a favorable environment for fungi would be somewhere warm and somewhere damp. Another example is budding in sponges, yeasts, and hydra. So this little organism up here at the top is a hydra, and you can see this is the adult here, and the adult hydra has 3 buds growing on it. Well, there may be another little one right here. So there may be 4 buds growing on this hydra. Once the buds get large enough, they will then pinch off and attach to some type of substrate like a rock or something and become their own organism. So that's budding. Again, one parent, they're going to be identical, so it's asexual. And then finally, this is probably my favorite example, is regeneration in planarians. Now, planarian is this little guy down here on the bottom left. This is a diagram of a planarian. It is a type of flatworm, and they have remarkable regeneration properties. So what that means is if you cut him in half, then the left half will grow a right and the right half will grow a left, and you'll get 2 planarians. If you cut him in half this way, the head will grow a tail, the tail will grow a head, you'll get 2 planarians. You can even chop them into 3 pieces and get 3 planarians. When I was in college, we did an experiment with planarians where we would split just the head, and we were able to create a planarian with 2 heads. And then on one of them, I was able to split one of those heads and get a 3-headed planarian. So it's kind of cool how much of a regeneration property that they have. But again, one parent, so it's asexual reproduction, and all organisms must reproduce in some way. And number 5, life evolves. This simply means that life changes, and organisms will begin to adapt to the changes in their environment as a population over a long period of time. So, what is an adaptation? An adaptation is an inherited trait or characteristic or behavior that enables an organism to survive and reproduce successfully in its environment. For example, hummingbirds. Hummingbirds have this long, slender beak, and that beak is very well suited for obtaining nectar in tubular flowers like you see in the picture. You would need a very long beak to reach down into the bottom of those flowers and obtain that nectar. So he has a good adaptation for that. However, the hummingbird does not have a good adaptation for cracking open seeds. Okay, so his beak is not suited for that. So he has a good adaptation for this environment for obtaining food, while other birds may have beaks that are well suited for their environment and the type of food sources that they ingest. There are 2 important concepts when we talk about how life changes over time. Number 1, populations produce more offspring than can survive. So not all of the organisms that are reproduced are going to be successful in their environment. They are going to grow, and they are going to be able to hunt for food or go through photosynthesis if they're autotrophic. But at some point, some resource is going to become limited. So it might be food, it might be sunlight, it might be space. And then only those that are best suited for obtaining that food or sunlight or space are going to be the ones that reproduce. Some of them are going to die out. And so organisms within these types of populations will compete for those resources in order to survive. You may have heard of survival of the fittest. This means that organisms with the best adaptations for their environment will be the ones that are likely to survive, and they're going to reproduce. But what kind of organisms are they going to reproduce? They're going to reproduce organisms with those same beneficial characteristics. Why? Because they're going to pass down their genes. They have the genes or the genetic makeup for that characteristic, so they're going to pass those down to their offspring. So they're going to reproduce more of the same. So we see that over time, population might shift from a group that has one certain characteristic over to having a different characteristic. Secondly, mutations are found in all populations, and so no 2 organisms are exactly the same, because some mutations may have occurred in one of those organisms that didn't occur in one of the others. So we see mutations being passed down to their offspring as well, and that can indicate or cause differences within populations. This brings us to the concept of natural selection. Natural selection is the enhanced reproductive success of certain individuals within a population based on their inherited characteristics. This is one mechanism of the bigger idea of evolution, which is a change in the genetic makeup of a population over multiple generations. It's important to note that we are not referring to the evolutionary process as one organism morphing into another organism. We're talking about a whole population, remember, a group of species changing the characteristics over time. Like we see some characteristics more often, we see some characteristics less often. To give you an example of this, let's look at these grasshoppers. The grasshoppers here, some of them are green and some of them are brown. Which one do you think has the better adaptation for the environment that they're in? I would say the green. Why? Because they are blending in with the grass. They're camouflaged. So if a bird flies over, he's going to be able to see the brown grasshoppers a lot more easily than the green ones. So he's probably going to feed on more of the brown grasshoppers. So if that's true, then over time, we're going to see this population of grasshoppers, the brown color is going to decrease and the green color is going to increase. And this is what we mean by natural selection. The green is the better adaptation for the environment. We're not seeing a green grasshopper morph into a brown or a brown morph into a green. We're just seeing changes in the amounts of each of those that we're seeing in the population. Okay, so those are the characteristics of life. Excuse me, and the next thing that we're going to talk about is scientific method. Now, you've studied this before, so hopefully some of this will come back to you as we go through it. But this is just a general way of using evidence to answer questions and to test ideas. It's often described as a list of steps, although I like to think of it more as a thought process rather than a strict group of steps that we have to follow. The first thing that happens when we're wanting to figure something out is we ask a question or we make an observation. We may see something that we don't understand. We may see an organism that we've never seen before. So we make an observation on what we either see, hear, touch, taste, or smell. For an example, let's say one Monday morning, a teacher arrives to class to find that the classroom is too warm. She then asks the question, why is it so warm in the classroom? Okay, so she is making an observation. She has sensed that it is warm in the classroom and she's wondering what's going on. Why is it so warm in the classroom? So she's going to form a hypothesis. A hypothesis is a tentative explanation of the observation. Now, a true hypothesis has to be something we can test. It has to be testable. And it is best written as an if-then statement. So the teacher decides, if I turn on the air conditioning, then the classroom will not be too warm. So this is her hypothesis. She thinks that if she turns the air on, then it's going to get cooler in the classroom. Written as an if-then statement. Notice, if I turn on the air, then the classroom will not be too warm. The third thing that would happen would be experimentation. So in that case, she's going to turn on the air conditioning and she's going to see if the classroom temperature gets cooler. So the temperature in that case would be considered data. Data, it may come from observations in the natural world or from a controlled experiment, which we will talk about later. It can be qualitative or it can be quantitative. Now, qualitative, things like, oh, it's red, what color it is, or it is cool to the touch, or I see smoke coming out of it. Quantitative is going to be like numbers. We're going to get a specific temperature of the room, for example. Once we have done the experiment, we're going to analyze the data that we collected. So we're going to look for patterns in the data, and we're going to see if the data supports the hypothesis that we made or if it refutes it. In the event that the hypothesis is supported, we may want to do more experiments. We might want to repeat the experiments to get more data to support the hypothesis. But if the hypothesis is refuted, we may want to revise our hypothesis, come up with a new one, and then do some different experimentation. So this leads us to a conclusion. A conclusion evaluates the hypothesis. My hypothesis was correct because, and here's the data that proves it. My hypothesis was incorrect because, and here's the data that proves it. So when we talk about controlled experimentation, it's important that we conduct the experiment properly. And this brings us to experimental design. Now, an experiment is defined as an investigation carried out in controlled conditions. This means we are going to monitor the conditions of the environment so that we don't get any outside influence that would cause a problem to our data. The sample size is the number of individuals assigned to each group. So let's say we're doing an experiment to test the effects of fertilizer on tomato plants. The number of tomato plants that we test is going to be our sample size. Is it more believable? Is my data more believable if I tested it on 2 tomato plants or if I tested it on 200 tomato plants? My data is going to be more reliable the more times I've tested it. So the larger the sample size, the more reliable our data is. Third, we need to know about variables. Variables are elements of an experiment that can change. And some of those variables we want to change ourselves. That's what we're testing. For example, with the fertilizer in the tomato plants, we're going to give each group of tomato plants a different fertilizer. We're going to give this one Miracle Grow. We're going to give another one a generic brand. We're going to give another one, say, Triple 13. So we're going to give them different types of fertilizer. That is the factor that we are changing or manipulating, and so this is called the independent variable. I remember the independent variable is the one that I control. Okay, the investigator controls that variable. Now, what happens as a result of that independent variable is called the dependent variable. So the dependent variable depends on the independent. This is the response to the independent. We might count the number of tomatoes that we get from each of the plants on different fertilizers, or we may look at how tall the plants grew. Whatever our measurement is going to be when we're measuring the success of the fertilizers, that's going to be our dependent variable. Now, it'll be important that you can pick out independent and dependent variables out of experiments, and there is a practice worksheet in Canvas that you can use to practice those. You'll also need to be able to pick out a control group. Now, the control group does not receive the independent variable, does not receive the variable that we are testing. So we can use this as a comparison group. For example, in the tomato plant scenario, we would give one group no fertilizer. So not only can we compare Miracle Grow to generic to Triple 13, but we can compare Miracle Grow to no fertilizer. We can compare Triple 13 to no fertilizer, and so forth. So the control group is the one that does not receive the independent variable. The placebo, when we use humans for experimentation, if we're testing something on humans like a new medication, then humans have we have minds, and we can think and we form opinions. So we may let bias influence the results. So to prevent that, we would use placebos. These are inert or inactive substances that will resemble the treatment given to the experimental group or the group that actually gets a medication. So what do I mean by this? Well, one group in our study would get the new medication. The other group in the study would get, like a sugar pill. It doesn't have any of the medicine in it. That's called a placebo. And we would not tell which group gets what. So they don't have a preconceived notion of how they're going to feel afterwards. And our data is less biased. Another example. CJ wanted to see if listening to music would make the basketball players make more baskets. On day 1, he didn't play any music and counted how many baskets they could make in 10 minutes. On day 2, he played rap music and counted the baskets. And on day 3, he played classical music and counted the baskets. What is the independent variable? Well, remember, the independent variable is the one that the experimenter changes. What did CJ change each time he did this experiment? He changed the type of music. So in this case, the independent variable is the type of music. Let's see if I can get something that writes here. One day it was no music, one day it was rap music, and then one day it was classical. Next, what is the dependent variable? Well, remember, the dependent variable is how we're going to measure the effects of the independent. Typically, this is going to be some type of measurement. And what is he measuring here? The number of baskets that they made. So that should be a direct result of the type of music in this experiment. What is the control group? The control group, remember, is the group that does not receive the variable being tested. So if we're testing music, then this is going to be the group that did not get any music, and that's what he did on day 1. So that was the day with no music. And this is going to give him something to compare all the other days to. Now, this last one you may have to think about. What are some constants that should be considered? So constants are things that we want to keep the same between the groups so that our data is not influenced by that. We only test one variable at a time in an experiment. And so this time we are testing the effects of music. So music is the only thing we want to be different between the groups. What are some things that would need to be regulated to keep this fair from day 1 to day 2 to day 3? Well, for one thing, you would probably want to use the same basketball players. Obviously, some of us are better at basketball than others. I am not. I'm not an athlete. So you want to have the same ability level each day. So I would recommend using the same basketball players each day. Another thing would be the height of the basketball goal. You want it to be regulation height. Another one would be what did, I don't know, maybe the basketball players have for breakfast that morning. Okay, you want them to all, like if one of them's starving, the other one had a big protein-filled breakfast, and he might be on his game better than another one. So these are some examples of constants that you could consider. You want to use the same type of basketball with the same amount of air in it, and so forth. So you could get picky and you could really go all out with that. But these are things that may influence our results, so we want them to be kept the same. Okay, we're going to pause there on our lecture for this unit, and we will start with classification in part 2.